This is the second part of a lecture on bacterial conjugation from April 8th, 2020. And the focus of this lecture is to outline the mechanism of F-factor mediated conjugation and then compare F-factor mediated conjugation with HFR and F-prime conjugation. And so F-factor mediated conjugation, or F plus times F minus mating, happens between a bacterial donor cell that contains F factor, or an F positive cell, and a recipient cell that does not contain F factor, or an F negative cell. And so to start the conjugation process, genes that are encoded on the F factor in the F positive donor cell will generate the sex pilus, or this direct contact between the F positive and the F negative cell. And once that contact is established, this sex pilus can then contract and basically draw that F minus or F negative cell closer to the F positive one in order to make the transfer of DNA through the pilus a little bit easier. And so once the F positive donor cell and the F negative recipient cell are within a very close range, the sex pilus is shortened. The type 4 secretion system can assemble around the sex pilus, and the plasmid, or the F factor, can begin to replicate. And there's a special type of replication mechanism that plasmids use during conjugation called rolling circle replication, but you don't have to know the mechanisms for that for the purposes of this class. And so the secretion system assembles after the sex pilus has brought the cells close together, and then the F plasmid itself is nicked or cut on one strand, and those two strands are allowed to separate, and that is done by a complex known as the relaxosome. And so that relaxosome has cut and basically started to separate that F factor plasmid. Coupling factor then brings this unwinding plasmid to the secretion system and the sex pilus and localizes it here, and then begins feeding that single-stranded piece of DNA through the sex pilus from the F positive cell into the F negative cell. And an enzyme called relaxase remains associated with the DNA through this process to keep this DNA unwound until the entire plasmid has been moved into the F negative recipient cell. And so once that piece of DNA has been threaded through completely, both the F factor in the donor and the F factor in the recipient cell need to finish replicating to become double-stranded again. And that recipient cell ends up as F positive because now this recipient cell contains an F plasmid. So what that looks like animated is you have an F plasmid in your donor cell or your F positive cell, which can build a sex pilus and bind to an F negative recipient cell, pull that F negative recipient closer. The plasmid can replicate via rolling circle replication and be threaded through that pilus by the type 4 secretion system. And then ultimately, both plasmids from the donor and recipient cell are replicated into double-stranded DNA, making that recipient cell F positive. And so there is another type of conjugation known as HFR conjugation. And an HFR cell, as compared to an F positive cell, is a little bit different. And so an F positive cell contains the F factor in an independent plasmid. But an HFR cell contains this F factor plasmid integrated into the bacterial genome. And so you can see that up here in this HFR cell, which has a blue chromosome and then that F factor in red integrated within it. And the mating for HFR conjugation still happens between an F positive or plasma that contains or a cell that contains the F factor, and an F negative cell, which does not have that F plasmid. And so similar to an F positive cell, HFR cells are able to make a sex pilus, draw the F negative cell close to them, assemble the secretion machinery, and begin to replicate the DNA and feed it through the sex pilus into the recipient cell. However, during this process, the host cell or the donor cell doesn't actually have enough time to replicate the entire bacterial chromosome, which is a lot bigger than a tiny F plasmid. Meaning that before the entire replication or transfer of the whole chromosome is complete, the sex pilus breaks, and you're left with a small piece of donor DNA in the F negative cell, but it doesn't contain the entire F factor. And so at the end of HFR conjugation, the recipient cell remains F negative. It doesn't have an F plasmid that it can now then use to perform conjugation itself. But what can happen with these fragments from the HFR cells that are now in recipients is that genomic DNA that was transferred can be integrated into the host cell genome, or it can be degraded altogether. And so this is not particularly good method of making F positive cells, right? Because the recipient cells don't obtain the ability to be F positive or to perform conjugation, but it does allow for some mixing of genomic DNA from the donor cell into a host cell, and that can in itself increase genetic variation. And so to remind you how HFR conjugation works, you have the donor cell's chromosome and a separate F plasmid. That F plasmid is integrated into the donor cell's chromosome to make an HFR cell, which can make a sex pilus and bring this F minus recipient cell closer. The donor cell's chromosome will replicate and start to pass through the pilus, but not entirely. And that little piece can integrate itself into the donor cell, leaving you with an HFR cell and an F negative recipient. Right, so you can see the sex pilus here, the F plasmid integrating into the genome of the donor cell, and then that sex pilus binding to an F negative recipient and starting to transfer that F factor and DNA into the recipient. There's just not enough time for it to happen. And so you've got a little piece of chromosome from this original cell that can occasionally be integrated into the recipient cell, which remains F negative. And the last type of conjugation that I'm going to talk about is F prime conjugation. And this is also a mating between an HFR cell, or at least starts with an HFR cell, and then that is mated to an F negative cell. And so, as I said before, there are plasmids known as episomes that can exist both in the bacterial chromosome and outside of it. And there is a way to excise or cut out the F factor plasmid from the bacterial genome once it's been integrated. So those HFR cells have F factor integrated into their bacterial chromosome, as you can see here, right? So the chromosome is in blue and the F factor plasmid that's integrated is in red. But there is a way to cut out that F plasmid through a process called excision. And sometimes when that happens, you get not only the F factor plasmid, which you can see in red, but a little bit of extra from the bacterial chromosome itself, which you can see in green. And so when the F factor gets cut out of a bacterial host genome in the HFR cell, but some of that bacterial chromosome goes with it, like this green piece, it's no longer called an F plasmid, it's called an F prime plasmid, because it has everything that the F factor has and a little bit extra. And so the host cell or the donor cell becomes F prime. And that F prime cell can perform conjugation and mate with an F negative cell in the same way an F positive cell could. Because F prime plasmid contains all the same things as an F positive cell and just a little bit of extra DNA from the bacterial chromosome. And so it can generate a sex pills, it can pull the F negative cell towards it, and then in the same way as an F positive cell, the F prime plasmid can be replicated through rolling circle replication and transferred into the donor cell, transferred into the recipient cell, which can then become F prime as well. And so rather than an F positive cell generating another F positive cell, in the case of F prime conjugation, an F prime cell that's donating its plasmid to an F negative turns that F negative recipient into F